in our institute, and he's an isotope geochemist and geologist. I'm trying to understand how Earth systems interact with each other through the geological past. Take it from here.
Right. This is turned on now. Is this too loud? You good? Thank you, Neta, and thank you for coming. I'm going to present to you today a study that came out of my group, and specifically Michal, who was, I guess you all know her, Michal Ben Israel, who used to be a postdoc in my group. This is her COVID project. And it was recently published with our collaborators. Oh, sorry about that. Collaborators, so we have Rob Holder and Andrew Halliday Clark, who are geochronologists, and then Emily Smith and Lyle Nelson, who are sedimentologists. So I'm going to be saying a lot of we in this talk. When I say we, imagine that we is mostly Michal. Okay. So it is generally accepted that concentrations, like large concentrations of oxygen, molecular oxygen in the atmosphere, are a sign of life. But the question I want to be ask you today and try to get an answer is how much oxygen, molecular oxygen, is required for complex life forms to evolve, aerobic life forms like animals. And this is not just a interesting hypothetical exercise. This is important for the search of life in extraterrestrial planets. Because right now we have two telescopes in orbit that, and most importantly the new one, the James Webb Telescope, that systematically analyze the composition of atmospheres in remote planets, in exoplanets, and get spectra that looks like this. Now here they detected water in the atmosphere. But it's possible that in the near future, or sometime in the future, we are going to learn that a planet has a detectable, measurable amounts of oxygen in the atmosphere. What does it mean? Like, let's say we find 5% of oxygen. Does it mean that there's life, and how evolved are these life can be? To answer these questions, we need to understand the dynamics. Sorry about that. The dynamics between evolution and the composition of the atmosphere. And right now, we only have one place that we can do that. That is planet Earth in the geological past. Right? So what do we know about the evolution of life? Well, we know that life has been mostly single cellular and very simple through most of Earth's history. This is time in billion years. And then a lot has happened during the Proterozoic and mostly during the late Proterozoic. The first fossil that is maybe an animal is dated to 890 million years ago and is thought to be a fossil of a sponge, an early sponge. But this sponge was pretty lonely for, was a lonely one for a long time. But then towards the very end of the Proterozoic, during the period called the Ediacaran, many animals and many branches in the animal tree came to be. So we have the Dickinsonia, which is maybe an ancient jellyfish, we don't know. The first mollusks, arthropods, and vertebrates all came to be during the late Proterozoic and the beginning of the Paleozoic. Right? So a period also known as the Cambrian explosion. So this is the Cambrian explosion. What were the level of oxygen in Earth's atmosphere, and most importantly in the habitats, in the marine habitats in which these animals evolved, is a different question. We know that today there's abundant oxygen in the atmosphere. There's more than 20% of it. And the modern oceans are oxic, with the exception of few of very localized oxygen minimum zones. And when we go back in time and try to reconstruct the schedule in which oxygen was rising in the atmosphere, we have to rely on proxies, and specifically redox proxies, right? Redox-sensitive proxies. So here's an example of two such proxies. These are the concentrations of uranium and molybdenum in black shells. Both of them are oxyanions, so they tend to be enriched in the water when the environment is oxic, and depleted. They are particle reactive in reducing conditions, so they are missing from water in reducing environments. And we look at the record, and we see that during during the transition toward the end of the Proterozoic at around 0.6, 0.7 billion years ago, they shoot up, right? They increase dramatically, which has been interpreted as a sign for the oxygenation of the ocean. It was also termed the NOE, short for a Neoproterozoic oxygenation event, which may suggest that the first animals that evolved during this period, right, have done so in maybe oxic oceans. And maybe the evolution was even driven. Maybe it points to a causality between the oxygenation and evolution. In the plot here, what you will see is basically there's a bunch of constraints on the levels of oxygen. So we know that according to these records, marine anoxia was sustained until the very end of until the end of the Proterozoic, and then the oceans could have been oxic. So gray zones, I can't tell if you can see. I can see it perfectly. You can see that there are gray zones and white zones here, but white zones are permitted and gray zones are forbidden. Right? We know that these animals are aerobic. They require some oxygen. Right? So there must have been few percent, 2%, maybe less of oxygen to sustain these animals, but it could have been much more. But there are other redox proxies, and they tell a different story. So what you see here are, it's a compilation. It looks like very few, like 5 data points, but in fact, this is based on close to 4,000 data points that are reflect the speciation of iron, again, in shells, mostly in shells, coming from different marine environments. And what it tells us is that there's a lot of iron, of reduced iron in shells that extends all the way to the Cambrian, so well into the Paleozoic. And the only time where we see less of it, so more oxidized iron, is in the Ordovician and later, which suggests that marine anoxia maybe have continued into the Paleozoic, and maybe some of the animals evolved in oxic oceans, and the rest in oceans. But the beginning happened in oceans that were much more depleted with respect to oxygen. And this is not the end. We have more records. I bet that you can't read the axis here, so I will tell you that this is a reconstruction of of the rise of oxygen in shallow marine environments based on cerium anomaly. Cerium is another redox-sensitive element, and which shows a very sharp rise in the level of oxygen from few percent to modern values at around 400 million years ago, so well into the Paleozoic, right? Suggesting that maybe these animals or earlier animals evolved in oceans that were mostly suboxic. They had few. Sorry, this is annoying. They had few percents of oxygen, enough for them to cross the functional threshold for evolution, but overall the oceans had very little oxygen in them, right? So there's an uncertainty here, or a data gap regarding what is the schedule for oxygenation in this critical interval of time when animals evolved. And the reason for these different reconstruction, or the reasons, must come from some combination of either these proxies record different environments in the ocean, in an ocean that is very dynamic with respect to how much oxygen is in there. We can have one shallow marine environment that is nearly anoxic, and another one that is oxic existing at the same time. So this is one possibility. But also we need to remember that these redox sensitive elements are measured in rocks that are super old. And another explanation is that some of these records may be altered during the protracted time they span in the subsurface. And I'm going to focus now on the second explanation. So this is how rocks usually look like when they are 1 billion years old. They are messed up, right? There's veins and they are altered, right? And a good way to think about it is, what we're after here is a signal that comes from the surface, right? And if you think about the history experienced by rocks as a thermal history, which temperature increases and then decreases over time, then we are after a signal that is recorded here when the sediments are fresh. But then it gets altered during diagenesis and epigenesis and even metamorphism, and it can keep on altering all the way until we get the sample and put it in the mass spec to analyze. Another way to think about it is that as a sample goes from the surface to depth and back to the surface, it goes from, it recorded some signal of oxygen, initially at the surface, and then it will naturally enter a zone that has very little oxygen in it, that is the interior of the crust, and come back to the surface at the present, when there's abundant oxygen. And this last stage and processes that are associated with alteration at modern or nearly modern surface environment, like weathering that happens in modern atmosphere, can imprint these rocks with basically a false positive oxygenation signal, right? And this problem is basically haunting this field and has been acknowledged very nicely by Noah Planowski in a paper from 2020. Okay, so what are we going to do about it? We're going to develop a new proxy that is, first of all, new, so it's always good to have more proxies, but more importantly, it's going to be more resistant to alteration, and specifically to this final stage of exhumation and from depth. And it's going to be based on carbonate minerals, and carbonate minerals are great because they initially precipitate from the surface, at the surface, right? So as they do so, they will take different major elements like magnesium and calcium and oxygen from the water, but also elements that are interesting because they are redox sensitive, like uranium and lead. So we take it all in. And we are going to be focusing on carbonate mineral rock and rocks that are very abundant in the geological records, that are dolomites. So carbonates are great, right? Well, it turns out that carbonates are also horrible because they are very sensitive to alteration, and especially at depth. And what you see here, just to exemplify that, is a histogram that I showed you last time I gave a talk here about 5 years ago, showing the formation temperature of dolomites. This is just frequency, and based on clump isotope thermometry. And what you can see is that the Mesozoic and Paleozoic dolomites are all very hot. They are hot because they tend to alter in deep crustal environments. Right? So this is not great. This is exactly what we're trying to avoid here. And yet, what I'm going to show you here is that when we use not just measurements of uranium and lead, but uranium-lead dating of carbonates, we can get a signal that is carried from the surface, through the subsurface, and back to the surface of oxygenation. And I'm going to show you a reconstruction of uranium to lead ratios of dolomite precursory minerals, so the minerals that initially formed at or near the surface that would suggest an increase in the levels of oxygen in marine habitats that happened after 400 million years. And then we're going to talk a little bit about what does it mean for evolution of life on Earth and other planets. Right, so this is going to be a quick, not so quick, but a primer to uranium lead dating. So just to let everybody are on the same page, uranium lead dating is based on the decay of two uranium isotopes, uranium-238 and 235, two lead isotopes, 206 and 207 respectively. And we like to think about this system in the composition space that we call the Terra-Wasserburg. And on the y-axis, there's the ratio of daughters, 207, lead 207 to lead 206, and on the x-axis, we have the ratio of parents to daughter, uranium-238 to lead 206. So now let's imagine that we have a new mineral that just precipitated out of water. It's a carbonate mineral, but it can be any mineral. Right here. We can imagine this mineral as two compartments. One of them is pure lead that got into the mineral, and the other one is pure uranium. And if we just look at the pure lead component on this space, it will plot here. It has 0 uranium in it and some 207 to 206 ratio. This is the initial lead of this mineral. And then if we focus on the uranium compartment, well, over time, this uranium is going to decay to radiogenic lead. And this compartment will evolve over time along this trend, also called the Concordia curve. So at 50 million years, it would be here, and 100 million years, it would be here. At 4 billion years, it would be here. And now let's imagine that we take a bunch of minerals that have different uranium and lead ratios, and they all formed at the same time, and then we measure them after some time. And these minerals are expected to plot along a trend that reflect a mixing between these two compartments, between the initial or common lead compartment and some point along the Concordia that reflect the time over which they evolved. Right? It's pretty simple. So we can, and the difference between a mineral that plots down here and up here is how much uranium, or what was the original uranium to lead ratio in these minerals. So minerals that had a lot of uranium and, compared to lead will plot down here, and minerals that have more lead and less uranium will plot up here. Right? This is the mineral that has only lead in it. Right? So we can pass a trend through this line, and then we get 2 for 1. We get a date that is the lower intercept of this trend with the Concordia, and we get an information about the isotope composition of the initial lead in this system. Right? This is great. Right? And the way we actually do these measurements, well, we can do them in bulk. This is how we were done in the past, but now we can do them using laser ablation. So we take thin sections, thick sections, and then we identify dolomite fabrics in them that are associated with a single generation, so we think they formed at the same time, and then we ablate them. We analyze uranium and lead isotopes from spots in them, and if we're lucky, we get a figure that looks like this. We get an initial lead and an age, and this is, you can imagine, the best isochron that dolomite probably ever produced. Most of them look much worse, but they are still meaningful. So you're going to see very large arrow bars. It means that most or many of the analyses don't look quite like this. Right. So let's look at some dolomite. So let's get to know the samples. We analyzed samples that came from 2 basins in the US, so the Death Valley and Colorado Plateau. There's a total of 65 samples in this project, and the rest of them are just compiled. And they cover a period of time between the mid and late Proterozoic. So the oldest sample, the stratigraphic age, is like 1.2 billion years. To the Mesozoic. The youngest one, I think, are Upper Cretaceous. And samples have different types of fabric. Some of them are, all of them are replaced, dolomites that are associated with replacement. And some of them are massive. That means that they come from massive beds like this. This is the Redwood Formation at the Colorado Plateau. In thin section, it will look like this. Others are associated with surface deposits, right? So we have dolomitized, these are ooids that were rolled on an ancient seabed, and they are replaced, and they can be preserved fabrics very nicely. Also, dolomitized fossils are considered surface deposits. And other dolomites, a third type, are associated with microbial activity, microbial mediations. These are dolomites that are associated with stromatolites, or here, these are alternations, and I don't know if you can tell, but alternations of dolomite and gypsum with these very nice ondulations that are associated with microbial mats and microbial activity. Right, so now we have the samples, and I think we're ready to look at some data. And what we're going to see here is the uranium to lead date of these dolomites compared with the stratigraphic age, independently determined stratigraphic age. And if what happened is that these dolomites have formed at the surface and the uranium lead system remained closed since nothing else happened, we expect them to plot on a 1-to-1 line. And they don't. They really don't. Most of them are just too young. We have uranium lead ages that are that plot below the 1-to-1 line. Sometimes they are younger by hundreds of millions of years. Other times, like tens of millions of years. There are few or several points here that appear to be older. The only explanation that I can think of is that these samples contain some contribution from detrital, an ancient detrital component. Think clay minerals that are very old in the system. And so I'm going to these are these samples. Right, so they're too young. And now we're going to look at the second parameter. We're going to be looking at the initial 207 to 206 in the samples and comparing them with the uranium lead date of these samples. If these dolomites have formed at the surface and nothing happened since, we expect them to follow this trend, which is the evolution trend for terrestrial Earth. Right? We expect the lead composition of the ocean to roughly follow this trend, and they don't. They all plot below this trend. And having lower than expected initial 207, 206, there are, again, some samples, 3 samples that diverge, that are plotting above this trend, but note that 2 of them are suspected for contribution from the detrital component. Okay, so it's a systematic observation. Right, so the question now is why? Why are they too young and have too low of an initial 207, 206? So Michal and I considered different scenarios that can explain this phenomena, and we came up with this one. We think it's pretty robust. The idea is that all of these minerals started off at the surface, taking uranium, taking lead, and then maturing as time went by and they got buried. Right? So they had an initial that was sitting on this trend, this terrestrial evolution trend, and a slope, and at that time they gave the right time, the right initial. But then, at death, they got altered, and they did so in a closed system with respect to lead. So imagine that we take lead from all of these samples after some of them evolved, some of them didn't, and now we homogenize it, right? So we turn these two compartments, the initial lead and the radiogenic lead, into a new one that is then also give us the new initial. Then we get a new initial that is lower, right? And then we let the system evolve as a closed system, right? Over, let's say, another 150 years. So in this process, during the homogenization, we get a new initial, but also reset the age to 0, right? This will intercept the Concordia at infinity, which is equivalent to an age of 0. So now we go and measure this Dolomite, and we end up with an age that is too young, right? It's 150 million years instead of 180. That is the stratigraphic age of this Dolomite, and an initial value that is too low, right? What it tells us is that first of all, is that these Dolomites have all undergone, or many of them, most of them, undergone alteration at depth. This can be associated with dolomitization, or more likely dolomite crystallization, because dolomitization involves very large water-to-rock ratios and is likely to reset the lead composition back to that of in the ocean. And recrystallization is more of a closed system process. Okay, let's take this a step further and think about what controls the lead the deviation of initial lead from this the original initial. Well, two things. First of all, how much time has passed between mineral crystallization and alteration. The more time the time will pass, the steeper this slope will be, and the lower the average 207 to 206 will be, right? And the second thing is, what is the initial or average of the initial uranium-238 to lead-206 in the system? Because if this ratio is higher on average, then the homogenization value will be lower. And we know how much time has passed between deposition and alteration, right? We know the stratigraphic age, we know the uranium-to-lead age. So we can calculate what was the initial uranium-to-lead ratio in the precursor, in the dolomite precursory mineral, right? So this is going to be our proxy. And this proxy is great because when you think about it, we have a uranium-to-lead ratio that is initially recorded here, and then the mineral is getting buried to some extent. We have some radiogenic lead that is evolving in the system in proportion to how much uranium is in it. And then the system becomes homogenized. And now we have a new initial lead. And this initial lead is resistant to alteration. It won't respond to later oxygenation, right? So the signal is now carried to the surface. And uranium at this point can change. I don't care about it. The initial lead is carried to the surface without alteration, and specifically without experiencing this false positive oxygenation signal. Right? So this is going to be a resistant redox proxy, an alteration-resistant redox proxy. Okay, so next thing to do is let's calculate what is the initial uranium to lead, uranium-238 to lead-206 ratio in these samples, and we did that. And this is what we get. We get, and note that this is the reconstructed 238 to 206 on a log scale, right? So there's a huge signal here. And values remain pretty low until 400 million years ago when they go up, okay? So this is interesting. What can explain this? First of all, maybe it doesn't have to do anything with oxygenation. Maybe this is just a signal of reflecting uranium to lead partition into different carbonate precursory minerals, right? Aragonite tends to take more uranium than a calcite, for instance. But when you compare these records to reconstructions that show what was the abundance of aragonite and carbonate, which mineral was more abundant in the ocean, we find that there's no relation. You see times where there's aragonite forming and there's very high concentrations of very high initial values of uranium to lead, and others that they don't. Same thing with calcite. So this is not a good explanation for the main trend here. So we are left with a different explanation that what controls this trend is really the availability of uranium and lead in the water from which these precursory minerals were forming. And what will control the uranium to lead ratio is, of course, how much oxygen is in the system. So when water are oxic, like in a modern ocean, the uranium to lead ratio is very high. This is because, as I told you, uranium tends to go into solution. Lead is associated with iron, iron geochemistry, and will tend to be particle reactive and removed from the system. And in reducing environments, we get the opposite, right? So high uranium to lead ratio is reflecting an oxidizing environment, or oxic environment. But where? Like, is this, maybe this is just maybe this reflects the entire, the global ocean, or maybe a region like, continental shelf, or like an entire marine environment. But we need to remember that we can get redox profiles in sediment. So maybe this just reflects, the formation of these precursory minerals at different locations in a sediment profile. To answer this question, what we did here, is to plot or color code the dolomites by their origin, microbial or surface deposit or massive. And if this is all the signal that was forming in the sediment, we expect that microbial dolomites that are associated with very sharp gradients of redox in the sediment will systematically be lower. And yeah, maybe they are a little bit lower here, but remember, this is a log scale. They are significantly higher than anything that we find here. The main signal that we see is not controlled by local variation in redox, and we associate it with a larger scale oxygenation. Maybe at a scale of an environment like shallow seas, and maybe even global, right? I would say that redox in the sediment can be a good explanation for the large scatter we see in values. So it can really explain well why we took a bunch of samples and one plotted here and the other one plotted here. Right? This is a good way to scatter the samples, but it can't explain this the overall trend. Right, and so we accept now that this reflects a rise in the levels of oxygen of marine environments, marine habitats, and this rise comes much later, like between 1 and 200 million years after the radiation of metazoan or animal, I should say, life and the time of the NOE. When we compare it to more recent reconstructions of the rise of oxygen on Earth, these are based on modeling of geocarb, also on constraints that come from redox-sensitive elements. It seems to follow very nicely this trend. Okay, but I don't want to be overexcited here. You need to remember that this proxy, right, the uranium to lead, doesn't give us a quantitative estimation of how much oxygen is in the environment. It's just a qualitative one telling us when the oceans became more oxic. when we compare this, we can compare this trend to other redox proxies that were sampled. This is the Cerium anomaly one that was also sampled in carbonates in the shallow marine environments, showing an increase at around 400 million years. And also a record here of iron speciation in weathered oceanic crust. This is a work of Stolper and Keller from deep ocean basins, right? When we see oceanic crust, that shows a similar step or increase that is very late into the Paleozoic. So all of these together support that this signal is A, global one, and B, that oxygenation came very late it came very late. Right, so what can we learn from it? So what does it tell us about the environments in which the first animal emerged? It tells us that these animals have emerged in oceans that were mostly limited in oxygen. They must have some, because these animals or these organisms are aerobic. They respire oxygen. But experiments that looked at, let's say, how much oxygen a sponge needs, or animals that are bottom feeders at the oxygen minimum zones, tell us that very little oxygen is sufficient for these animals to thrive. It takes something like 0.2 to 2%, and you can have animals living in the oxygen minimum zone. And so this is one scenario. The oceans are overall oxygen limited. Another possibility is that we can't rule out is, yes, maybe animal life emerged in oceans that were more oxic. But if they were more oxic, it happened over very short intervals, right? Or very local oxygenation, like oxycoesis, that did not register in all of the redox sensitive proxies I just showed you, right? This is also a possibility. Right, so I think this is the last question I'm going to ask. Okay, so what finally has driven this increase at 400 million years? Why the levels of oxygen in the environment were going up just then? So generally speaking, if you want more oxygen in the atmosphere and the ocean, what you really want to do is to get photosynthetic organic matter and bury it. Right? Just producing it is not enough, because if you produce organic material, it will get oxidized. And you leave it to the surface, it will get oxidized and consume all the oxygen. If you want to see more oxygen in the atmosphere, you need to bury it. And two mechanisms come to mind that are associated with the late Paleozoic and later times. One of them is the expansion of vascular plants, basically trees, that came about at 400 million years or so, which A, increased the biomass on Earth by a lot, and B, delivered a lot of organic material to be buried in basins. And the second one that we can consider are improvement to the biological carbon pump. So, as I told you, what you want to do is to bury organic material. And one way to bury more organic material is to pack it better. For instance, by having larger mass of organisms, right? Organisms are larger. This is a more efficient way to reducing power from the surface of the ocean to the sediment, right? So turns out that here the expansion of land plants happened just before or in the very early stages of this very sharp increase that we're seeing. Right? I still have one slide to go after that. So just to conclude, we've seen that uranium, I hope I convinced you that the uranium-led system in dolomites, but very possibly in other minerals that are orthogenic and are forming in the ocean, can carry signals of oxygenation from the surface through burial and exhumation. And then the reconstruction of uranium to lead ratios based on this method show us that the levels of oxygen in ancient marine habitat increased dramatically at around 400 million years ago. This timeline suggests that early animal life evolved in basins that were, or in oceans that were largely suboxic. They had very little oxygen in them, maybe just enough to enable these early animals to respire. So you can imagine like very low metabolism, very slow moving animals. Or maybe, they were moving faster, but during very short intervals of time and very localized oxygenation events. And finally, we think that the final or oxygenation, persistent oxygenation of the ocean after 400 million years is likely reflects or was possibly driven by evolutionary steps like the expansion of vascular plants of trees. So what is the lesson that we learn from an N=1 planet? Is oxygen a sign of life? To the first order, yes. Right, to have more oxygen in the atmosphere, the mechanism that we know of is having a lot of photosynthesis. And but we can have complex life forms like animals, aerobic life form, evolving in environment, thriving in environments that have relatively low oxygen in them. Maybe a few percent is enough. And with that, I am done. Thank you.
Questions?
I think it's a fascinating work, and you squeezed a lot of information from playing with the uranium lead system. I just wonder, why did you choose the Terra-Wasserberg plot? Because I think an ordinary plot of the people use always lead versus the uranium ratios and using U values and
so like a single decay chain.
Yes.
Like the
combination of the two. You need to
Yeah.
combine the two systems. But there are other options. I mean, did you try with other methods of, or you just went on with this one, and since it was satisfactory, you stayed with it?
So honestly, I didn't. This is just the Terra-Wasserberg plot is a convenient way of crossing these two systems and looking at them at one go. And it's really a convention in this, uranium lead dating of carbonates. This is what they use. It would work. This approach would work in any way you choose to display. So this is just a method of basically presenting the data. We have an analytical derivation of this equation. It would work on any way you want to display it. Yeah.
So there's evidence for oxygen in the atmosphere 2.4 billion years ago.
Oh yeah.
So how comes the oxygen in the ocean stayed so low for such long time after the atmosphere already oxidized?
So the oxygen in the atmosphere was rising in steps. The first step is really which I didn't talk about, is the great oxidation event at 2.4, 2.3 billion years ago. The great oxidation event, just to put in perspective what is great, is the atmosphere increasing, the levels of oxygen in the atmosphere going above 1 per mil. Yes. This is what it takes for the sulfur non-mass dependent anomalies to be gone, right? To have enough of ozone layer to block the UV light. So you can have the oceans remaining and the atmosphere remaining largely anoxic, right? throughout most of the Proterozoic and even well into the Paleozoic.
Do you think you can calibrate the uranium and lead ratio to like 0 oxygen? Like you can convert this ratio to how much oxygen you have. You can calibrate it to an oxic environment.
Yeah. So this would take a lot more expert. So I wish I could present to you something that is a quantitative calculation of how much oxygen is in the environment based on this proxy. But it would take a lot of experiments and probably field studies. And also this can be very much complicated by the speciation of uranium and lead during redox, and preferential maybe uptake of different uranium species. So this is a big question. So right now I can't. So, but yeah, good point.
I have a question.
Yeah.
So, like a preliminary thing that you need to use this is that the alteration will be in a closed system for lead, right?
Yes.
Would it be the case with other minerals? Or, yeah.
It might or it might not. I can tell you that it seems that calcite is also being recrystallized, often recrystallized at depth, and the two hallmarks of closed system recrystallization are too young of an age and too low of initial lead. We see that also in some calcites, apatites as well, bioapatites.
Iron oxides?
I'm sorry?
Iron oxides?
Maybe. I don't know. Could be. Yeah, that'll be an interesting target. One of the problems is that not all of these elements are, well, to measure, to get uranium lead dates, you need to be able to standardize your measurement. And this is, the people that are the geochronologists that do the analytical part of these projects tell me that it's not as easy as